Welcome to Catalyst MedHub, where we ignite understanding and make learning easier. In our today's class, we shall be discussing on fertilization, cleavage, and blastocyst formation. It is an important embryological process which leads to the development of the human. The first one is fertilization. Fertilization simply refers to as a process in which the male and the female gamete fuse together to form zygote. It usually occurs in the ampulla of the uterine tube or in the ampulla of the fallopian tube. Fertilization occurs within 24 hours after ovulation. Now, but before the fertilization to occur, there are things we should take note of, or there are things that are very important for us to know, such as the movement of the sperm cells, the ability of the sperm cell to break the barriers that surround the oocyte, and also the reactions that occur between the sperm cells and the barriers that surround the oocyte. The first one is the motility or the movement of the sperm cell. During sexual intercourse, about 200 to 300 million sperms are deposited into the female vagina, but it is only 200 to 300 sperm cells survive to reach the site of fertilization. The movement of the sperm cell from the cervix to the isthmus of the fallopian tube takes about 2 to 7 hours, and this movement is facilitated by the prostaglandins that are present in the semen in which the sperms are suspended. Also, the movement of the sperm cell is also aided by their protoplastic movement. After the sperm have reached the isthmus of the fallopian or uterine tube, it will lose its motility, or the rate at which it will move decreases until after the egg have been removed, or until during ovulation. During ovulation, when the egg is being released from the ovary, the egg will move with the aid of its cilia and the contraction of the smooth muscles into the ampulla of the uterine tube. Now, once the egg is in the ampulla of the uterine tube, it will now be releasing a chemical attractant which will attract the sperm cells which are already in the isthmus of the uterine tube. Then the sperm cell will now move towards the egg that is in the ampulla of the uterine tube through the chemical attractant that is released by the egg. So after the sperm have reached here, the sperm have to break barriers before it can enter the egg, or before the fertilization should occur. And the barriers include: 1, the corona radiator, 2, zona pellucida, and 3, vertebral membrane. These 3 barriers are very important for the sperms to penetrate before fertilization should occur. Now, for the sperm to penetrate these 3 barriers, it must undergo some conformational changes in the female genital tract, which is termed as capacitation and acrosomal reaction. First one is capacitation. Then the second one is acrosomal reaction. Capacitation is the process in which the glycoprotein, the vesicular plasma protein covering the cell membrane, are dissolved from the acrosomal region of the sperm. And it is only the capacitated sperm have ability to penetrate the egg before fertilization to occur. So also, capacitation simply refers to as the conformational changes in the female genital tract. So that is the capacitation. The main essence of the capacitation is just to dissolve the glycoprotein and the seminal plasma protein that's covering the cell membrane of the acrosomal part of the sperm cell, so that the acrosomal enzyme will be viable. The next one is acrosomal reaction. Acrosomal reaction simply is the reaction between the digestive enzymes that are present in acrosomal cap of the enzyme with the zona pellucida. That is among the zona pellucida, which is among the barrier in which the sperm must penetrate before fertilization to occur. So those are the major things we need to take note before fertilization. The next is the steps of fertilization. Steps of fertilization. The steps or stage of fertilization include 1. Penetration of corona radiata. 2. Penetration of zona pellucida. 3. Fusion of sperm and oocyte membrane. 4. Completion of second meiotic division. Completion of second meiotic division and completion of second meiotic division and formation of female pronucleus and formation of female pronucleus. And 5. Formation of male pronucleus. And lastly, the 6th one is formation of zygote. So these are the steps or stages of fertilization: penetration of corona radiata, penetration of zona pellucida, fusion of the sperm and oocyte membrane, completion of second meiotic division and formation of the female pronucleus, formation of the male pronucleus, and lastly formation of the zygote. The first one is the penetration of the corona radiata. Only capacitated sperm cells have ability to penetrate the corona radiata. So any sperm cell that have been capacitated, it will penetrate the corona radiata. Then from here, penetration of the zona pellucida. The sperm cell that penetrate the corona radiata will now come in contact with the zona pellucida. And in this process, the sperm cell will now release its digestive enzymes that are present in the acrosomal end of it, such as the acrosin and trypsin-like enzymes. This acrosin and trypsin-like enzyme will digest the zona pellucida, which will now make a way for the sperm cell to enter into the oocyte. Now, after the sperm cell have already penetrated the zona pellucida, there will be a reaction. There will be a conformational changes in the property of the zona pellucida called zonal reaction. This zonal reaction will make the zona pellucida to be impermeable for other sperm cells. And the zonal reaction is being initiated or is being carried out in the presence of the zonal proteins. The zonal proteins bring about the zonal reaction, and it involves the dissolution of the zona pellucida by the acrosomal enzymes such as acrosin or hyaluronidase or a trypsin-like enzymes. Next one is the fusion of the sperm and the oocyte membrane. Once the sperm and the oocyte membrane comes in contact, they will now diffuse. After that, the head and the tail of the sperm will now penetrate the cytoplasm of the oocyte. Once the head and the tail of the sperm penetrate the cytoplasm, there will be a calcium barrier formed around the cytoplasm, which will make it impermeable for another sperm to penetrate. Next one is the completion of the second meiotic division and formation of the female pronucleus. The oocyte is in the metaphase of second meiotic division, that is during puberty. Now, once the sperm cells comes in contact with the oocyte will now complete its second meiotic division. Then, after that, it will now give rise to two daughter cells, the large cell and the small cell. The large cell is the one with high content of the cytoplasm, and the smaller cell is the one with little content of the cytoplasm called the polar body. The large cell will now develop and form female pronucleus. The next one is the formation of the male pronucleus. Now, the sperm cell will now move towards the female pronucleus, and its nucleus become enlarged. After the nucleus become enlarged, it will now shade up its tails and become male pronucleus. The last one is the formation of the zygote, which is the essence of fertilization. Now, the male and the female pronucleus, they are now in contact with each other. Their cell membrane dissolve. Then after the membrane is dissolved, their chromosome will now come in contact with one another, which will lead to the reaction. Then at this point, the diploid form of the chromosome have already been restored, because the male pronucleus has to degree, the female pronucleus has to degree, their reaction together will now form the diploid, and at that point the zygote is already formed. So that is also the step of fertilization. This diagram here summarizes the steps of the fertilization. The first diagram here is showing us how the sperm penetrates the coronal radiation. So this is penetration of coronal radiator. So we see that the capacitated sperm will be the one that will penetrate the coronal radiator. So this is the coronal radiator, and this is the sperm. These are sperm cells that are trying to penetrate the coronal radiator. So after penetrating the coronal radiator, next is the penetration of the zona pellucida. This is penetration of zona pellucida. Penetration of zona pellucida. Here now, this is the sperm. This is the sperm cell trying to penetrate the zona pellucida. This is our zona pellucida. I would say that it's brought about by the releasing of its acrosomal enzyme, which dissolves the zona pellucida. Then after that, the sperm cell will now fuse with the membrane of the oocyte. So here is the fusion. Here is the fusion of sperm cell with oocyte membrane. The sperm cell will try to fuse with the membrane of the oocyte. Then here, the sperm cell have already penetrated the cell membrane. I will say that it is the head and the tail that will enter the membrane, as well as the mitochondrial sheath. They will be left at the surface. So here is entry of sperm cells in the oocyte membrane. The next one here now is the formation of the pronucleus, both male and female pronuclei. Inside here we are having the male pronucleus and the female pronucleus, and with the formation of the polar bodies. So these are the polar bodies. The next step here is the dissolution of the membrane of both male and female pronuclei. Now here the membrane have already dissolved, which lead to the mixing of their chromosomes content. Here now the chromosomes will now mix together. That is, we'll be moving from haploid to diploid, because the male pronucleus contain haploid number of chromosomes, the female nucleus contain haploid number of chromosomes. Once they come in contact, they now form diploid number of chromosomes. And lastly, the chromatin of the zygote will now arrange themselves in the equator. After the arrangement in the equator, then the zygote is formed. So here is our zygote. These are the steps that are involved in fertilization. The next thing is the result of fertilization. Results of fertilization. What are the results of fertilization? The results of fertilization include 1. Restoration of the diploid number of chromosomes. That is 1. To restore diploid number of chromosomes, that is from haploid to diploid. Another result of the fertilization again is initiation of cleavage. initiation of cleavage. That is a series of mitotic division in which the zygote undergo. Another one again is determination of the sex of the zygote, because during fertilization the sex of the zygote will be known. If the female pronucleus contains X chromosome and the X sperm fertilizes, it will result in the formation of XX. And if you are having XX as the chromosome, that is a girl child. And if the female chromosome is fertilized with Y sperm, we will be having XY, which corresponds to the male child. So determination of the sex of the zygote is also part of the result of the fertilization. So determination of the sex of the zygote is also part of the result of the fertilization. So the result of fertilization includes the restoration of different number of chromosomes from the haploid number, initiation of the cleavage, and determination of the sex of the zygote. That is all on the fertilization. The next thing is the cleavage. Cleavage is just a series of the mitotic division in which the zygote undergo. Cleavage usually begins 30 hours after fertilization, and it occurs as the zygote is moving from the ampulla of the uterine tube into the uterine cavity. So as the zygote is moving from the ampulla of the uterine tube or fallopian tube into the uterine cavity for the implantation, then the zygote will be undergoing a series of mitotic division called cleavage. And the pattern of the cleavage includes this zygote that will form after fertilization will be the one that will undergo cleavage. So from here, this is the cleavage spindle. As the zygote is moving from the ampulla to the uterine cavity, it will be undergo a series of mitotic division called cleavage. And the resultant cells that are formed in each cleavage, they are called blastomeres. Blastomeres. Those are the cells that are formed during cleavage. Now, the zygote will undergo mitotic division and give rise to two cells, which is called two-cell stage. So these are our two-cell stage. Two-cell stage. It will give rise to two cells, the larger cell and the smaller cell. So from here we can see this is the larger cell and this is the smaller cell, and both of them they are surrounded by zona pellucida. Now, after the two-cell stage, the larger cell will divide mitotically to form the three-cell stage. So here, this is the three-cell stage. The larger cell will divide first to form the three-cell stage, also covered by zonal pellucida. Then, after the three-cell stage, the smaller cell that have not divided from the second cell stage will also divide mitotically to form two, which will result in the formation of four-cell stage. So these four-cell stage. All these cells here, they are called blastomeres, and as they keep dividing, the more they will be becoming smaller. As they are dividing, they will be becoming smaller. But the entire size of the embryo will not change due to the presence of the zona pellucida. So for the four-cell stage, this is the zona pellucida. Now, the next one is that the four-cell stage will also divide to form eight-cell stage. The eight-cell stage will further divide mitotically to form 16-cell stage, which resemble a mulberry. Hence, it is called Morula. So, it is 16-cell stage called Morula. Because it resembles a mulberry, it is called Morula. So the 16th cell stage of the cleavage is called Morula. then the morula will keep moving, still surrounded by the zona pellucida. Zona pellucida. Still surrounded by the zona pellucida. The morula is formed at about 4 days after fertilization, and at about 5th day after fertilization, the morula have already entered the uterine cavity. And as it's moving to the uterine cavity, the process of the formation of the blastocyst will begin. Because these morula cells, they will arrange themselves into 2 layers. The cell of the outer mass layer and the cell of the inner mass layer. The cell of the inner mass layer, they will be the one that will form embryo. So they are called embryoblast. Whereby the cell of the outer mass layer, they will be called trophoblast. So from here, this outer mass cell, and inside is the inner mass cell. As the morula enters the uterine cavity, the fluid from the uterine cavity will now begin to penetrate the zona pellucida and fill the spaces, intercellular spaces of the morula cells. And from there, the small cavities will begin to appear, which will later fuse together to form a large cavity, and that cavity is called blastocyst. From here, this is our trophoblast, and this is the inner cell mass which is called embryoblast. And from here is the fluid that enters the cell from the endometrium of the uterine cavity. Now next, this small cavity will now begin to fuse together and form a large cavity which is called blastocyst. So this large cavity here is called blastocyst. And at this stage, the embryo or the morula will now be called blastocyst. So this entire structure is now the blastocyst. Now the inner cell mass, which is the embryoblast, will now move to one pool called the embryonic pool. And as the fluid keep accommodating in the blastocyst cavity, the zona pellucida will dissolve. And after the zona pellucida dissolved, then the blastocyst is ready for implantation. This blastocyst is filled with a fluid that is rich with nutrients. It is also called the uterine milk. The main reason, or the major important reason why the zona pellucida keeps following the zygote from the ampulla of the uterine tube up to the uterine cavity is because other cell has high ability to attach to the wall of the uterine tube even before reaching the uterine cavity. So the zona pellucida will just prevent the implantation at any region outside the uterine cavity. So once the zona pellucida dissolves in the uterine cavity, the implantation will now begin, and that is about 6 to 7 days after fertilization. And that is about 5 to 6 days after fertilization. So this is about the formation of the blastocyst. Once the endometrial fluid fills the intercellular space of the morula and the cavity is formed called the blastocyst, at that stage the morula or the embryo will be called blastocyst. That is all on the fertilization, cleavage, and the blastocyst formation. Kindly subscribe, like, comment, and share. Don't forget to turn the notification bell so that you don't miss any new video.